Goldfish exhibit a wide range of morphological variations. In previous videos, we explained how these differences arise due to distinct genetic backgrounds among different strains. For example, if the coding gene undergoes a mutation, the goldfish develop twin tail, whereas without mutation, it remains a single tail. Additionally, genes responsible for eye shape and fin length have also been identified. The factors contributing to this morphological diversity include gene duplication, nucleotide substitutions, and artificial selection by humans. In this video, we will examine the diverse mutations observed in goldfish and analyze their morphological evolution from the perspective of evolutionary developmental biology. By the end of this video, we will understand why developmental biology is essential for understanding the mechanism underlying the morphological variation in goldfish. Furthermore, we will gain a deeper appreciation of the significance of conducting evolutionary developmental biology research using goldfish as a model organism. Now let us begin. We have discussed in previous videos, goldfish have a unique genetic background that includes gene duplication and a high tendency for genetic mutations. Because of this, one might argue that goldfish exhibit a wide variety of phenotypes simply because they are prone to genetic mutation. Indeed, the fact that genetic mutations occur frequently helps explain why diverse phenotypes can emerge. However, this raises an important question. Does the mere occurrence of genetic mutations mean that the more mutations occur, the more morphological variations will arise? We have previously compared the goldfish and the common carp koi, specifically considering why twin-tail goldfish exist while twin-tail koi do not. Through this comparison, we have come to understand that an organism's body does not immediately accept a genetic mutation and translate it into a new morphological form. This suggests that there are inherent limitations to morphological variations in goldfish as well. The same principle applies to goldfish. In other words, the diversity of goldfish strains also has certain limitations. In this series, we examined research findings on the intriguing relationship between the tail and the dorsal fins of ranchu goldfish. The findings suggest that producing a stable strain of ranchu-like goldfish with no dorsal fin and only a single tail fin is quite difficult. This is because without a mutation in the coding gene which causes the tail fin to bifurcate into two, the dorsal fin tend to retain. This implies that the goldfish strains do not arise in as many variations as most of us expect. But rather than that, there are specific morphological forms that are inherently difficult to achieve. At this point, you may have started to realize that discussing genetic mutations alone does not fully explain why certain goldfish shapes and strains do not exist in the marketplace. If you still believe that everything can be explained by random genetic mutations in the genome, consider the following question. Why don't two-tailed goldfish become an established strain? In fact, even in our own laboratory, we sometimes observe goldfish babies with two heads. However, there has never been a successful case where such goldfish have become an established strain. If your thought is that they are too abnormal and weak to survive, so they are naturally eliminated, then you are on the right track. Let's take it one step further. If a fish only had a slightly unusual shape but was just weak, does it seem like it could survive with careful human care? In reality, twin-tail goldfish and ranchu appear more delicate than wild-type goldfish. However, breeders can raise them with care, allowing them to grow into adult and produce the next generation. This suggests that we need a more detailed explanation for why extremely unusual morphological variations in goldfish, such as the two-headed goldfish, fail to become an established strain. When a specific gene undergoes a mutation, a certain phenotype appears, and that phenotype becomes subject to selection. This fundamental principle applies universally to organisms, from microorganisms to vertebrates, regardless of body size or complexity. However, what I want to emphasize here is how the genotype actually translates into the phenotype. I believe that this process varies depending on factors such as an organism's size, number of cells, and structural complexity. To be precise, this is not an idea I came up with on my own. It has been a topic of discussion among many researchers in the past. To better understand this concept, let's take a look at this diagram. This diagram was not originally designed to explain the relationship between genotype and phenotype. However, it provides valuable insights for understanding this discussion. Let's consider the case of unicellular organisms like bacteria. These organisms reproduce by cell division, passing their genetic material directly to the next generation. As a result, they do not through the long developmental process that we typically associate with growing from an egg into an adult. In contrast, organisms like goldfish and humans are multicellular and reproduce through sexual reproduction. This means that males and females produce sperms and eggs, which must meet and fertilize to create a new organism. That fertilized egg must undergo a long and complex developmental process before becoming a mature individual capable of reproduction. The adult body is much larger than the fertilized eggs and consists of specialized tissues and organs. As a result, the developmental process takes significantly longer. I believe you would all agree with this point. Now let's consider a specific gene as an example. One that we have frequently discussed in this video is coding gene. Whether or not this gene undergoes a mutation determines whether a goldfish develops a twin tail or remains single tail. However, at the moment of fertilization, there is no tail in either single tail or twin tail goldfish. Therefore, whether or not the coding gene has mutated at this stage makes no immediate difference in the tail phenotype. This does not mean the coding gene is inactive during early development. Its effects are not yet clearly visible to us. The actual impact of this gene mutation only becomes apparent much later in development. This concept applies to many other genes as well. In multicellular organisms with complex body structures, specialized cells divided and differentiated into various tissues and organs. This process involves long and intricate development. Because of this, the simple equation of genetic mutation equal phenotype does not always hold true. When developmental processes are involved, the relationship between genotype and phenotype can be represented using a diagram like this. In this diagram, the lower section represents the genetic space, the upper section represents the phenotype, and in between there is a conceptual layer called the developmental space. This illustrates how genetic information is ultimately translated into the final phenotype through the developmental process. However, applying this diagram directly to our goldfish example can be somewhat challenging. Terms like genetic space, developmental space, and phenotypic space may seem too abstract to easily connected to concrete examples. So let's consider a more practical example that is easier to visualize in order to better understanding this concept. Among the examples we have discussed in this video, the rancho goldfish provides a relatively clear illustration of this concept. So let's use ranchu as a case study to explore the relationship between genes and phenotypes. In previous videos and this one, we explained that the coding gene which causes the twin tail fin is also related to the presence or absence of the dorsal fin. Why does the coding gene affect both the dorsal and the tail fins? The reason originates in early development. During the initial stage, these two structures are positioned very close to each other. A single gene may function to closely related regions during early development. However, as the cell divides and migrates over time, these regions become distinctive, resulting in different phenotypes in different locations. This highlights that the relationship between genes and phenotypes is not a simple one-to-one correspondence. Instead, it must be understood within the context of developmental time and spatial organization. The principle we have discussed regarding to the coding gene apply not only to coding but also the other genes. For example, when explaining the development of twin-tailed goldfish, we mentioned the gene that determines the ventral side. Multiple genes are expressed on the ventral side, including the BMP gene. The name BMP represents bone morphogenetic protein. Initially, a member of the BMP gene family was discovered to be involved in bone formation. However, later research revealed that BMP plays a role in various other developmental processes beyond just bone formation. From these examples, the principal insight is clear. A single gene is not necessarily confined to a singular function. Instead, genes often participate in multiple biological processes influencing different aspects of development in an organism. We will use this diagram to further explain the complex relationship between multiple genes and multiple phenotypic traits. This diagram is also not originally my own. It is based on the figure created by researchers. These researchers recognized early on that genes and phenotypes do not have a simple one-to-one relationship. In this diagram, the lower square represents the genotype while the upper square represents individual traits that constitute the overall phenotype. To help clarify this concept, let's apply this diagram to our ranchu goldfish example. The coding gene in goldfish influences both the shape of the caudal fin and the presence or absence of the dorsal fin. A more refined version of this diagram represents multiple genes as G1 and G2, demonstrating how they collectively contribute to higher order phenotypic traits. Next, let's use this diagram to explain how transition from single-tail to twin-tail variants occurs. As a fish, swimming ability is crucial for goldfish. At the same time, they must maintain essential physiological functions such as heartbeat and metabolism. Now let's consider what happened when coding gene loses its function. As we discussed earlier, when coding is lost, the caudal fin bifurcates into two. As a result, swimming ability is significantly reduced. Additionally, an important physiological trait related to survival might also be affected. If the impact on the physiological trait is not lethal, the fish can still survive. Even if its swimming ability is impaired, the fish could survive as long as someone takes care of it and provides food. If a twin-tail variant is visually appealing to breeders and fanciers, artificial selection may allow this phenotype to persist. Through this explanation, you can see how these diagrams help illustrate real examples of goldfish morphology. However, let me add a brief clarification here. The examples we've discussed so far are explanations based on observed facts. In reality, many more genes are involved and the actual processes are far more complex than what we have described. It's important to keep this in mind when considering genetic mechanisms. That said, the key point here is that a single gene can influence multiple phenotypes. Furthermore, these phenotypic traits are interconnected and influence each other. The more complex organisms, the stronger these interrelationships become. When an organism's development and growth span a long period, it remains exposed to environmental influences for an extended time. The embryonic development process is highly regulated by precise molecular mechanisms forming essential structures such as eyes, mouth, fins, and muscles. However, external factors like temperature fluctuations or exposure to specific chemicals can significantly disrupt this process. Even after birth, environmental conditions continue to play a role. Whether the goldfish grows in an environment exposed to sunlight or whether its diet consists predominantly of animals-based or plant-based food can alter its final phenotype. Considering these factors, it becomes clear that the simple notion of genotype equals phenotypes is not enough. Additionally, not all mutations occurring in the genome show up in a way this visible to us. This becomes clear when we consider studies on laboratory-induced mutants. Numerous experiments have been conducted to investigate gene functions by introducing genetic modification in various organisms. Particularly invertebrate model organisms like mice and zebrafish, researchers have artificially introduced genetic mutations and analyzed their phenotypic effects. These experiments allow us to induce genetic mutations throughout the genome, including those that never occurred naturally during evolution or were eliminated by selective pressures. However, what about the result of such extensive mutation-inducing experiments? Were astonishing, extraordinary animals successfully created? Did mice or zebrafish with radically abnormal morphologies become stable laboratory strains? There are indeed cases where zebrafish with a twin-tail phenotype similar to that of goldfish have been artificially produced. However, we do not see stable lines of mice with two tails. Likewise, there are no reports of zebrafish with two heads swimming around in laboratory tanks. Some of you might conclude that extreme morphological mutations are too harmful. If you are thinking along these lines, you are on the right track. This insight will help us to understand the next part of the discussion. Not all genes carry the same level of importance. Some are absolutely essential for life, while others play less critical roles. Additionally, we have established that a single gene does not necessarily correspond to a single phenotype in a one-to-one manner. In some cases, a single gene may influence both crucial and relatively minor traits. If we examine the temporal and spatial aspects of development, our understanding of the many-to-many relationship between genotype and phenotype becomes more clearer. As we saw in the case of ranchu goldfish, cells originate from the same developmental region early in embryogenesis. Later, these cells can migrate and differentiate into entirely different organs or structures. Furthermore, the same gene can play different roles depending on the timing and location of its expression during development. Keeping these developmental biology principles in mind, let's further explore the intricate relationship between genes and phenotypes. So far, we have used the example of ranchu goldfish to illustrate how a single gene can influence multiple traits. This phenomenon, where one gene affects multiple phenotypes, is known as pleiotropy. For example, the coding gene not only causes the twin tail in ranchu goldfish but also plays a role in the loss of the dorsal fin. Similarly, the BMP gene is involved not only in the formation of the dorsal ventral axis but also in bone development. Many other genes also exhibit pleiotropy affecting multiple developmental processes. If a gene mutation disrupts a crucial function necessary for survival, the individual may not survive, making it unlikely for the mutation to pass on the next generation. For example, in goldfish or koi, a mutation may create a body shape that is visually appealing to humans. However, if that same mutation impairs cellular processes essential for the survival, it will not persist across generations. The effect of pleiotropy also vary depending on the time and the spatial organization of development. The longer developmental process and the more complex body structure, the stronger constraint imposed by pleiotropy. A longer developmental period means that even minor disruption during the process could prevent the organisms from surviving. Similarly, larger organisms with highly differentiated tissues and organs have many critical regions that cannot be easily altered without severe consequences. If a mutation occurs in a gene that controls multiple important traits, the chances of that individual surviving and reproducing decreases significantly. As a result, the range of the genetic variation that can be tolerated within a species is constrained. This suggests that extremely unusual or drastically different body forms are unlikely to emerge easily, imposing restriction on the direction of evolution. The constraints and evolutionary directions are not solely due to pleiotropy. There are several other factors that limit how traits can evolve. For instance, some pleiotropic traits are controlled by highly complex gene networks, making them difficult to modify. In such cases, the network's stability may be robust that small mutations in one or two genes do not lead to visible phenotypic changes. Additionally, multiple traits may be tightly interconnected, making it difficult to alter one trait independently of the others. In unicellular organisms, such a constraint may be less significant, allowing random mutations to create advantageous phenotypes that can survive and propagate. However, in more complex organisms like goldfish, such changes do not occur as easily. In theory, we introduce a vast number of random mutations and allow them to accumulate over a period. We might expect to freely create goldfish with ideal traits according to human preferences. However, if strong developmental constraints exist, then desirable traits may never emerge in the first place, preventing breeders from selecting them. This means that no matter how strong artificial selection is, there are fundamental limits to its effectiveness. Through the discussion so far, we have considered questions such as, why do not certain traits and varieties fail to appear? Why do some varieties emerge more easily than others? We have moved beyond the simple explanation of genetic mutation and the selection alone. It is now clear that developmental biology is essential. It helps us understanding the evolution of organisms with large multicellular and structurally complex bodies. By integrating insights from both developmental biology and evolutionary biology, we can analyze how the highly complex morphology of goldfish changes over time. This is one of the main reasons why I study evolutionary developmental biology through goldfish. Instead of simply linking mutation and artificial selection, I emphasize the importance of considering the developmental processes that mediate between them. This approach helps us understanding not only what types of evolution are possible, but also what types of constraint or difficult to achieve. In this regard, goldfish serve as an excellent research model. Now that we are in the latter part of this video, let's consider a new approach to evolutionary developmental biology in goldfish. In recent years, genome editing technology has been applied to various organisms. This powerful technology allows us to precisely modify specific regions of the genome at will. In fact, we have also conducted experiments using this technology to alter the genome region coding for the coding gene. And indeed, we were able to introduce mutations precisely where we intended. Previously, while discussing genome editing technology with a visitor to our lab, he asked, if we use genome editing technology, can we create designer goldfish just like designer babies? My answer was yes and no. Why? The reason is closely tied to everything we have discussed so far. Genome editing technology allows us to freely modify genetic sequences. However, even if we can change the genome sequence, our understanding of how these genes function during development is still incomplete. In other words, simply editing genes does not guarantee that we can create entirely new and extreme goldfish forms. So when asked if goldfish editing alone can produce unprecedented goldfish, we cannot simply answer with a yes. With current genome editing technology, we can likely achieve certain modifications such as changing colors, increasing or decreasing muscle, creating goldfish that resemble existing strains. These are examples of quantitative change to existing trait. However, what about radical transformations? Could we create creatures with fantastical forms like those seen in manga or fantasy? Could we design an entirely new form of goldfish, something that have never existed in the history of life? The reality is this remains extremely difficult. Take for example the two-headed goldfish seen at the beginning of this video. Could such goldfish grow into a fully mature adult capable of producing offspring? And could it be established as a stable strain? We still don't know. The key question is this limitation due to fundamental constraints in the genome and developmental process of goldfish? Or is it simply that our current knowledge and technology are still insufficient? Regardless, further research will gradually uncover the answer to these questions. Finally, let's consider the significance of studying evolutionary developmental biology of goldfish from a different perspective. Goldfish are multicellular organisms that reproduce sexually and have relatively large bodies. Furthermore, they are vertebrates with highly specialized organs and tissues. They have also been separated from their natural environment and subjected to artificial selection in controlled settings. Doesn't this sound similar to us humans? We too are large-bodied vertebrates with complex organs and tissues. We have continued to evolve through sexual reproduction. And now we live in an artificial environment increasingly detached from nature. Some researchers refer to this era as Anthropocene. Personally, I'm not entirely certain about the scientific validity or precise definition of this term. However, it is generally understood to describe an era in which human activities have significantly altered the Earth's environment and ecosystems. If that case, haven't goldfish already experienced their own Anthropocene? They have long been removed from their natural habitat and have lived under human management. In a way, they could be seen as pioneers in adapting to life under human influence. One of the unique aspects of goldfish is that we can closely observe their entire life cycle, from fertilization to embryonic development, from growth to adulthood. We can also study how human selection was influenced to their morphology over time. These processes can be analyzed in great deal. With this in mind, don't you think that through studying the evolutionary developmental biology of goldfish, we may gain insight into our own future form?